posted at Innovate Bio. Thanks, Cassandra. It'll be posted at innovatebio.org by Monday afternoon. Next week, oh no, this is our last AT project talk. Copy and pasting, always a bad thing to do. We're going to hear today about high school where they're doing DNA sequencing, which should be great. Joseph and Kissaou are going to be telling us about this. And each talk is going to be about 20 minutes long, and we'll have questions afterwards. At the end, we will have a survey that we'd really like you to complete. I will have the link in the chat, and this helps us with planning these kinds of events and figuring out how to best serve our community. All right, without ado, Joseph and Kissaou, we're looking forward to hearing about sequencing.
All right, well, thank you very much, Sandy, for the introduction. And I'd also like to thank Innovate Bio for hosting this project talk series. This is a great opportunity for Kissaou and I to share some information about our project. So my name is Joseph Oleniczak. I'm the principal investigator on the dual credit high school community college DNA sequencing and genomics project. I'm an adjunct professor at Austin Community College in the biotechnology department, and I'm also an instructor at the Liberal Arts and Science Academy High School in biotechnology. And I mostly deal with dual credit high school students in biotechnology. I'm joined today with my co-PI, Kissaou Tchedre, who is a member of the biology department at Austin Community College. And I guess we'll go ahead and jump right into our talk and talk about what we're doing with our project. But before we talk specifically about our project goals, I just want to sort of mention and give some context to what we do. Again, we're working on recruiting and getting more students into the ACC or Austin Community College Biotechnology program. And our overarching goal for ACC Biotechnology is to meet local workforce needs by increasing the number of students who are getting certificates and degrees from Austin Community College and joining the biotechnology workforce. Specifically, Austin Community College offers 3 different degrees and certificates: a level 1 certificate, which is sort of a fast track certificate that we've embedded in high school courses that I teach with dual credit students. They also offer a 2-year Associate of Applied Science degree in biotechnology. And lastly, they offer an advanced technical certificate, which is a post-baccalaureate certificate aimed at meeting the needs of students who already have a 4-year degree but want to get additional training in biotechnology. So today, for the dual credit portion that I'm going to talk about in our project, I'm mostly going to focus on this level 1 certificate because that's what I'm trying to get students in my program to complete. And we're doing that by encouraging them to join the program and increasing the enthusiasm through the program through some of the DNA sequencing projects that are supported by this project. Oops. So that level 1 certificate, just to kind of walk you through it, my students take the level 1 certificate over 2 years. They enter our program as 11th graders, and they complete all of these courses that are listed here. It's sort of an introductory course to college, the 1300. Then 2 biotechnology courses, one called Biology 1414, which is Introduction to Biotechnology 1. I teach that in the fall semester of their 11th grade year. And then in the spring semester of their 11th grade year, they take Biology 1415, which is Introduction to Biotechnology 2. Specific to this project, through funds through our NSF ATE project, we have embedded some DNA barcoding curriculum into Biology 1414. This curriculum was adapted from curriculum we received from our partners at the Cold Spring Harbor DNA Learning Center. And students do an introductory DNA barcoding project where they go out into nature and they collect a sample and they go through and barcode it using DNA sequencing. Then over the summer from 11th to 12th grade, they take a quality assurance in the biosciences course. And in year 2 of the program, they take a course called BITC 2486, where they do more extensive research using our sequencing facility established by this project, doing more extensive DNA barcoding projects. And I'll talk more about that here in just a moment. If you're unfamiliar with DNA barcoding, before I talk more about it, I just kind of want to walk you through at a very high level. Essentially what our students do in that 1414 course is they go out into nature and they collect a sample. So typically I have students who collect insect samples or plant samples. In this image here, we're showing a plant sample, and they prepare that sample by extracting DNA from the sample. Then they amplify it at a barcode region using PCR. They clean up that barcode, go through a cycle sequencing reaction, and then they run it through a sequencer we have on our high school campus to generate that barcode data. Lastly, they go through and they use bioinformatics to interpret that sequence data to identify the organism that they collected initially. So they do this as a group in 1414, and then they do more extensive barcoding projects in 2486, where they're looking at research questions like assessing biodiversity of a particular organism within a park in our area or around our school, or other applications of barcoding in that regard. As a result of our project, we've had a huge impact in the number of students who are completing the level 1 certificate. Our project kicked off in 2021, and we had 20 students that year who completed the level 1 certificate. Moving into year 2, as a result of our project, we had 41 students who completed the level 1 certificate. And next year, we're anticipating having 54 total students complete the level 1 certificate. In terms of recruitment numbers for biology 1414 and 1415, in 2021, year 1, we had 94 students. In year 2, we had 93 students. And next year, we're projected to have 136 students. This year 1 to year 2 sort of stayed the same because it was limited by the number of faculty we have on our dual credit campus, which is only me at this point. But next year, we're planning to add another person, which is why we can increase the number of sections of 1414 and 1415 and have more students move through our program. At ACC, in terms of 2 years traditional students, we started year 1 with 6 students. This year, we have 33 students, and we're projected to have 60 students next year. In terms of our project outcome, we basically had 3 key findings that we wanted to share with you guys. As a result of our project, we have had a significant increase in the number of students reporting skill level increase in DNA sequencing. We've also had a huge increase in the number of students who want to complete the level 1 certificate. And lastly, we have a huge increase in the number of students who actually want to go into biotechnology as a career. So again, students took the DNA barcoding module using our sequencing facility. Our sequencing facility has an applied biosystem Seq Studio genetic analyzer, where students can do single read analysis on DNA barcodes using that workflow that I mentioned earlier. Before they go through that curriculum, we have them take a pre-survey, and then after they have completed that curriculum, we have them take a post-survey. And on this slide, we're showing you pre-survey data over here on the left for panel A, and over here for B, we're showing post-survey data. We asked some questions such as, rate your understanding on how DNA sequencing works from no knowledge, beginner, proficient, advanced to expert. We then had them look at this or rate their understanding of DNA sequencing instrumentation, understanding of how to execute laboratory protocols for DNA sequencing, and finally, how to interpret DNA sequencing data. And you can see here in pre-survey results, most students rated themselves as having no knowledge or beginner knowledge. And then as a result of going through this curriculum, almost all of our students were in the proficient to advanced to expert category after going through the experience. The second key outcome we wanted to talk about is the number of students who are working towards the level 1 certificate. Again, pre-survey data is shown over here in A, and post-survey data is shown over here in B. We had a marked increase from the number of students who were interested in completing the level 1 from that pre-survey data to post-survey data as a direct result of going through the DNA sequencing module. And although it doesn't show it right here in just sort of these percentages, as a dual credit instructor who's taught these courses for 15 years now, I can really say that having the sequencer on campus has generated a whole lot of excitement in my biotech courses. And students, after they get an opportunity to work with this high-level instrumentation, get really excited about working in biotech and working towards this level 1 certificate. The other key outcome we wanted to show from our survey data was the impact on students who want to go into biotech or the biosciences as a career. Again, we're showing pre-survey data over here on the left and post-survey data over here on the right. And before going through the DNA sequencing module is sort of across the board how interested they were in pursuing a career in biotech or the biosciences. And after going through that DNA sequencing module, we saw a marked increase in the number of students who not only wanted to continue that level 1 certificate shown in the previous slide, but who actually wanted to go into a career in biotech. More specifically for you to hear this project, like I mentioned earlier, we have 54 students who plan to continue in the program next year to get that level 1 certificate. Next year, we've recruited 136 students on my campus who want to go through those 1414 and 1415 courses. I mentioned doing the more extensive DNA barcoding projects in 2486. This year, I piloted some techniques of having virtual mentors from industry work with groups of students who were in that 2486 course, and it worked really well. Basically, I had students come up with a DNA barcoding project that they wanted to work on, and then seek out a virtual mentor. So one example of that is I had a group of students who were looking at a local park and how ant biodiversity varied along a transect. And they reached out to some researchers at the University of Texas at Austin Brackenfield Field Lab who are specifically working on ant research, and had them mentor and sort of develop their research questions, as well as come up with some of their sampling techniques before they went out, sampled all these ants in a local park, and then used our sequencing facility to determine the species types for those different ants, as well as to look at that biodiversity assessment. Another thing I worked on this year is since our sequencing facility is automated Sanger sequencing for we want to start moving towards using NGS platforms to do next generation sequencing for metagenomics. So I partnered with a group from the University of Texas at Austin Freshman Research Initiative that's looking at bioprospecting enzymes from different microbes within superworms that degrade plastics. So in other words, we're growing superworms under different conditions, degrading plastic, and then collecting the bacteria from the microbiome of each one of those different setups, and looking at how that biome shifts over time by doing NGS metagenomics, looking at multiple reads in parallel. So that's one thing I'd like to move more into moving forward with this project into year 3 and then beyond the project. Now I'd like to pass it over to Kissaou so he can talk about this from the 2-year perspective.
Okay, thank you also for coming. So at ACC for the 2-year program, so we have also students who also are interested in level 1 certificate or advanced certificate if they already have a bachelor degree, and some of them just go for associate degree, and after they transfer to 4-year college to complete their degree. And I can tell you currently we have a collaboration with UT where our students, after they finish, for example, their associate degree, they are able to transfer their program to UT for 2 more years, and then they get their bachelor degree in biotech or biochemistry. So for a 2 years program at ACC in spring 2023, we had 18 students. This is one section, students. And then in the program, they always, we have a section where they have to run labs. And then this lab is the DNA barcoding, which runs through several weeks until they sequence their samples. So students are the one who collect samples. They bring the sample in class, they extract the DNA themselves, they amplify the coding region, they check the quality of their DNA samples, and then they sequence it. For my students, some of them have the chance to go to LASA at Joseph's facility to load the samples themselves to the sequencer. So for the fall, we had 20 students. This was 2 sections. And again, every semester, student for 1414, there is a module where they have to do the DNA barcoding. And at the end, they will analyze their data using a bioinformatics tool, which is a DNA subway developed by our partner in New York called Spring Harbor Laboratory. So it's a very important bioinformatics tool that students use to analyze their sequences. And then at the end, they can identify the plants, the barcodes. Okay. So starting from last spring, I also introduced the barcoding of moth and then algae. So students are, in addition to the plant sample they bring in, we give them also some moth samples or algae sample, and then they process all the sample together and then run it through the sequencer. So you can see that they have now a chance to run many samples, while in previous time we used to send samples outside for sequencing. So since we have everything in-house now, it's much easier for students to run more samples. So for the fall 2023, we project again about 20 students. And then for BITC 2024, which are internship students, so we expect about 24 from Joseph's program to train them for their internship. And after that, they should be able to complete their program. So also, I'll be teaching all those courses, those internship courses, using again the Genomics Center. Okay, for curriculums, as you can see, we added the DNA barcoding of 2 other species like moth and algae. We used to do in previous semesters only plants. Now we are expanding this to moth and then algae barcoding. So research students, they process all those samples and then sequence them. So where they got the moth samples or algae, moth samples come from a collaboration with Curtis, which is a professor and department chair of biology. So he teaches 1407, which is a biological organism. So he collects all those moth samples, trying to survey the environmental change in the Austin area due to constructions and so on. So he collects the sample, he takes the picture, he identify all those moths by their name, and then he sent us some samples for barcoding. So this is a collaboration, and then at the end, the student prepare the report and then send back to Curtis for his program. So we also plan for 1406 to add now this program in addition to 1414. We want to add also the same program to biology 1406. And this will allow us to increase again the number of enrollment. So toward this ends, currently biology faculties are learning how to barcode plant or moth, especially moths. And then with that, they will be able to incorporate that also in their program. So recruitment with our Genomics Center, as you can see the numbers shown by Joseph, we see some increase in number in enrollment and so on. And then we expect this number to continue. And hopefully we should be able to also get some more, another sequencer so that we can increase both the use of those sequencers at LASA and then at ACC. So this is going to be a plan for next year. So we also increase the number of minority students in both programs at LASA and then at ACC by directly talking to or attending events, minority events, or talking to some minority institutions around the Austin area. And we saw some significant increase in student enrollment. So that's what I have to talk on my side for the 2 years program. Joseph, you can move forward.
Thanks, Kissaou.
Sure, no problem.
We kind of wanted to end on showing some pictures of our students in action. So down below here, this is a picture of our lab at the Liberal Arts and Science Academy. Most of my sections of 1414 and 1415 have a total of 24 students. So if you're curious of what DNA barcoding looks like in a large lab facility, these are our students down here working. Over here, this is our sequencer and our sequencing facility. It's an applied biosystem Seq Studio genetic analyzer. And again, it's for automated Sanger sequencing for single reads. So these are some of my students asking questions before we load the sequencer. And over here, we actually have students loading the sequencer and programming the sequencer to collect their data. Lastly, down here on the far right, I mentioned the metagenomics project that we are working on with superworms with some partners at the University of Texas at Austin Freshman Research Initiatives. And these are students who are seeding their terrariums with the superworms in order to kick off those experiments. I also wanted to include some pictures that Kissaou is going to walk us through of his students when they came to LASA to run their samples on our sequencer.
Yes, you can see these are students. This just happened last week. So you can see students, they will process the samples at our campus, at the ACC campus. They process everything all the way until the samples are ready to be loaded to the sequencer. And then we take the sample with the students. They go over there, they finish the processing, and then they load it to the sequencer. And at the end, Joseph will upload all the files, the quality analysis results, everything, and send the files back to us. And then we proceed with the bioinformatics analysis of the data. So this is how we work together with between LASA and ACC.
And lastly, we just wanted to talk about moving forward. So we're in year 2 of year 3 of this project. And moving forward, we want to continue to recruit more students into our program. We really want to work towards sustainability. Running a sequencing facility is expensive. So we're considering starting a 501c3 where we can start to collect fees for sequencing from other local high schools, as well as other departments in Austin Community College. And then lastly, we want to continue to pilot some of these NGS metagenomic projects with the University of Texas and sort of think about moving our sequencing facility forward to include NGS platforms so we can start doing NGS analysis. And with that, I think I'm about out of time, so I'm going to stop sharing my screen. And I think there are some questions in the chat.
There are some questions, yes.
Right. I'm going to actually hold off on the questions until Dr. Kamajaya's talk is ended. So this is great. So we're going to, I'm going to introduce Erin now, and then we'll have questions for everybody after that. Okay, so now I'm happy to introduce Dr. Erin Kamajaya from Los Angeles Pierce College, and he is going to talk about a project where he has undergraduates, not undergraduates, I'm sorry, high school students do internships in his lab at LA Pierce College in biomanufacturing, learning how to manufacture all different kinds of programs. So Dr. Kamajaya.
Okay, I was trying to find where's my mic unmute buttons. Hi everyone, my name is Erin Kamajaya. I'm the PI for the ASPIRE project. This is the grand number. Can you see my pointer? Yes. Yeah.
Yes.
Okay, thank you. So this year is the first time we actually have our formal biotech training program. So in the very first part of the talk, I want to talk about what I learned, my experience in getting the first course, the first cohort up and running, and then I will give a brief sort of like update on the Taq polymerase that we produce, as well as other target expansion. So in case some of you who are not familiar, the ASPIRE, we received the funding to basically to start not only the biotech training program, but also biotech internship to produce some biotech product that can be useful for bioscience classroom. And we choose Taq polymerase as our product because that seems to be very commonly used in the classroom. If anybody teaching PCR, polymerase chain reaction, most likely you will need some Taq polymerase, and we'll be more than happy to share all of our protocol, even the product itself. We can send the Taq polymerase to your institution. Just make sure you let me know, and we can arrange something. Okay, so in the beginning, at the beginning part of the talk, I want to talk about our training program, what I learned from getting the first cohort running. Last year, if you attended the talk from me last year, basically I talked about how we had a high school student. We partnered up with one high school, Reseda High, to have the internship where students come to our facility and they involved in the hands-on activity, producing enzyme and media, things like that. We have that the internship every Saturday from 9 to 3 p.m. And the reason why is because that's the time where high school students are out of the school, right? That's the only time we can do that. And we are able to recruit a lot of high school, and this is just the number, 12 students in the fall and as many as 20 students in the spring. And that's because we have a very highly dedicated high school instructor that help us to recruit the student. So I think that's one of the sort of like, if anybody asks me what sort of the secret, what is the secret sauce to get a student in your program, I think you have to have a champion in on the institution of on your partner institution, I guess. So, but in the fall, when we arrive in the fall 2022, that's the beginning of our biotech training program. That's the we will start our biotech training program, and at that time due to the limitation of the sort of like space and classroom availability, at that time we only had we only have one choice to offer it at night, from 6 o'clock to 10 o'clock at night. And this is one of the things I learned. It was very hard for us to fill the class, and for whatever reason, right? So luckily for us, I had my summer student that allowed me to give sort of like survey what's the best time to host the class, right? And most of them indicate that they're available in the weekend, which is something I never thought about. But this is sort of like in a business we call that market research. You go to the customer and see what's the most convenient time for them, what's less barrier for them to join, and that seems to be Saturday, Sunday in the weekend. So we decided to have our biotech training program on Saturday, Sunday. This is the first time we have that, and in fact, we are the only class in our district, Los Angeles Community College District, that offer a class on Sunday, by the way. So, but because of that reason, because we have this class in Saturday, Sunday, unfortunately, we were unable to offer the internship for the fall 2022. So we put that one on hold. So how does that work in the weekend? Well, the moment we switch the schedule in the weekend, we double the amount of students who actually enroll in the class. And we were able to get full enrollment in, I think, a few weeks before the semester start. And this sort of like the profile, we started with full enrollment, 20 students registered in the course. And then, actually 18 of them stay after the census date, and then 17 completed the course, kind of like 85% completion, or if you count from the 18 student total, it's actually 94% completion. Basically, in the second course this semester, spring 2023, 16 of them are in the course right now. So this is like the first time we have this, and I was quite pleasantly surprised that we were able to fill the course, considering that this is the very first time we offer the program, right? That the question that I had at that time is where do these students come from, right? Which recruitment strategy actually helped us to get the student in. This is just a slide to summarize various effort that we've done that actually get able to attract the student. In summary, essentially, there's no one magic bullet in a way, right? So there's no one magic recipe. You do one thing and then your class is filled. It's not like that, but it's more like we do quite several different outreach activity, and from each of these activity, you get a few students here and there. For example, like we have the biotech internship from the first cohort, the one that I showed you in the second slide number 2. We got 4 students from that, right? And then we also have antibody hackathon event. We also have the Biotech, the bioscience club, Symbiotics over here, the workshop, as well as visiting classroom, flyer, social media, Instagram, things like that, and the exploration course. And all of these contribute some X number of students to fill the course. So that's sort of like what I found it interesting. So in terms of the class, here's just the structure of our class. We focus, our training program is industry focused, industry heavy, I guess, right? So 25% of the score coming from the documentation. We put heavy emphasis on the documentation because if things are not documented, it doesn't happen, right? So we really put that emphasis on that. We also have this laboratory technical skill, as well as the theoretical knowledge. This is mostly just laboratory math and some background information. And all of this in the theoretical knowledge here, we have this sort of like a questionnaire in the middle of the semester, as well as at the end of the semester. It's not multiple choice because we don't believe industry. We don't believe in multiple choice that much. Anyway. So and then we also put emphasis on the employability skill. In addition to all this, We also took our student to a local biotech incubator at Pasadena Bio Collaborative to be involved in supply chain activity, creating material for engine biotech experience. And also we train our student to do some sort of like root cause analysis when things go wrong. It's like this one here. From when we look at the learning outcome, I think all of the students reported improvement in the lab skill proficiency. So this is actually what you've seen over here is the pre-post comparison, or I should say the difference between pre-survey and the post-survey in terms of the skill at this different category, the lab skills, safety, ability to maintain the lab instrument and things like that, working as a team, documentation and all that. What's interesting is this is actually the survey conducted by our external evaluator. They actually separate the response between the students who have prior lab experience. These are the students who have taken majors biology or some sort of high school biology before with the lab component, and students who have 0 lab experience. So we have about 7 out of the 17 who participated in the survey, 7 of whom have 0 lab experience. This is actually non-STEM majors. So non-STEM students, they heard about biotech, they are interested, and they join our training program. And what's interesting, all of them reported an increase in all category. But what's more interesting when I see this is that students who have 0 lab experience, prior experience, reported a larger amount of increase in terms of the proficiency, particularly in the lab safety. So if you look at everything, yes, they have increased on the proper laboratory documentation, but the largest difference come from the lab safety here. So this is the one with no prior experience. So anyway, so that's what we learned from the first course, the first biotech course that we have. From then, some students continue to the second course, which is the we call it the Biotech 3 course. So in here, we try to focus on assessing the student, or at least giving the student opportunity to plan their experiment, to manage their time, and do something more than just learning technique, right? Because one thing that what I found is, particularly this is in my experience with my high school student, they're very bright students. So if you ask them, what is the theory behind PCR, they can tell you what it is. They may even be able to run the experiment themselves, but they may not be proficient in, let's say, planning. For example, like if you want to do protein purification, well, you need to make the buffer first. So oftentimes this kind of thing doesn't dawn on them that, hey, we need the buffer, right? So in the second course, what I envision is to give them this opportunity to plan their experiment themselves, and which is very hard to achieve in the standard classroom model where everybody is doing the same thing, right? When the entire class doing, okay, week 1 will do nickel his Taq purification or hydrophobic purification, it will be hard to achieve this kind of like having students to get this opportunity to plan for themselves. And this is why we decided to take sort of a risk, I guess, sort of like a new, different model of classroom instruction where every student will have a different project. So, but at the same time, everybody will have to know the core concept of biomanufacturing. So our second course here in Biotech 3, mostly in the focus on biomanufacturing, the student will have to learn the upstream processing and downstream processing, the component of each, the theory behind it, but all those material will be taught in a flipped classroom model kind of thing. So I have the pre-recorded video where they have to watch, and we will have a midterm questionnaire as well as the final questionnaire. But in the day-to-day classroom, they have to conduct their own project. like this. So they lead their own project. So most projects are industry focused project, like validation, method development, assay development, things like that. But we also have projects that are more academic, discovery based project, like the tiny earth, like the DNA barcoding, C phages, things like that. So we have sort of like different spectrum of project for student, and they get to choose which project they want. But while we're doing all this, I thought, hey, maybe it's time for me to start having or reintroduce the internship again. So I thought maybe we can pair the student who have no lab experience with my biotech student who already undergone through one biotech course in the fall, in the previous semester. With that thought, I thought, okay, now we can have a sort of like mentorship model, I guess, right? Where each biotech student will be a mentor to a student who have no prior research lab experience. So that's what we do this semester. It's kind of hectic right now because we have 15 biotech students, which serve as a mentor, and 15 biotech mentees. These are the students who have 0 lab experience. So we have a 30 students in the classroom every weekend, I guess. So that's very interesting experience. I'll be more than happy to share about that in my presentation next year, I guess. I'll tell you what happened then. But so far it's been very interesting dynamics, and I think the biotech student benefit from the experience on mentoring the new student, whereas the new student, the mentees, they also benefit to see what biotech lab is, to get a sense what the day-to-day operation, and they get working under the wing of the mentors. So I think that we'll see what happens. Our evaluator will look into this and conduct the evaluation, and I can share the result with you next year, I guess. So now, in terms of the science part, the update on the Taq polymerase as well as other target, what do our students do? So previously, we basically express and prepare the crude Taq polymerase extract. If you attend the NSF ATEPI conference, you probably have seen my student presented her and her colleagues project in the conference. So basically, we prepare the crude extract and we test it against the commercially available Taq polymerase. For this, we choose the NEB, and from there we sort of get a sense of what is the best, I guess, the best amount, the optimum amount of Taq polymerase that we should use for PCR, right? So you can see from here the 2T. I know the labeling may be a little bit need more explanation. The 2T, 1T, 0.5T, 0.25T. Essentially, if it says 1T, that simply means 1 microliter of our crude extract in 1 PCR reaction, 25 microliter PCR reaction. So if it's 2T, then you have double the amount of Taq extract, which is 2 microliter. Half T is half of the amount, and so on and so forth. So you can see from here we tested by amplifying the lambda DNA. We use the BABEX, Bay Area Bioscience Education Collaborative, a community protocol, because we know that works and it's very standardized. So we use that as our benchmark, I guess, to test our Taq polymerase against the commercially available one. And we see from here, hey, our 1 microliter dose is comparable to the NEB, to the commercially available Taq polymerase. Okay, yeah, so that's what we did in the past. So in terms of what we're doing right now, we try to optimize the lysis, the expression, and everything. For example, we set up our first our Sonication sort of like machine. So our student basically looking up the protocol, see what how do you set up Sonicator, you know, and then we tested with our Taq polymerase, and then from that we discovered that, of course, sort of expected, Sonication improved the yield, right? So you can see this one here, the one on the left is the amplification from using the sonicated sample, whereas the other one is the one we use the standard protocol from DNA Learning Center, which is just using Freeze-Thaw method to lyse the cell. So the Freeze-Thaw method is we don't dilute it, whereas the sonication we already have the 1 to 5 dilution, and then even then you know, we only need about half the dose, only half the compared to the Freeze-Thaw, right? So something like that. And the explanation for this, the most likely explanation is because we just have more cells that are lysed using sonication. So you can see from there, right? The sonication, you see more cells, more sort of like darker band, I guess, because a lot more cells are lysed during sonication instead of in comparison to Freeze-Thaw. But anyway, we also tried our crude extract with different kind of activity, including the DNA barcoding, the RBCL, and CO1 amplification, cytochrome C oxidase. We also tried it with the genotyping for the bitter tasting receptor. This is the TAS2R38. We also tried it also with the 16S ribosomal RNA barcoding, and we actually sent this after we amplified this, we send the sequence to DNA sequencing, and we get reasonable amplification all the way to 800 base pair. So I think our crude extract can be used, have a wide range of application. So that's good. So now what we're currently working on is to develop a lyophilization method. We tried that. This is our student who is the lead on the lyophilization. She created the matrix for various formulation. We do have a problem with our lyophilization machine, but luckily we reach out to our industry partner, and they allow us to enter their facility, and we learn from this scientist how to operate lyophilization. So again, we work very closely with our industry partner to provide a good training for our student. In addition to that, we are working on getting purified Taq polymerase. As if you remember from the gel I showed you earlier, it was just crude extract. So there are a lot of junk, a lot of the other protein in the mix. So our hope is to be able to purify that Taq polymerase. And then we also try to develop this sort of like automated protein expression using the CGQ and the LIS method. This is a work collaboration with actually we get this fellowship from Biomatech. So what you see over here is the cells grown in the flask. As you can see, that green light is the sort of like a way for this equipment to quantify bacterial growth. So instead of taking sample one by one and looking at it under with the spectrophotometer, you can quantify the bacterial growth just by looking at the amount of scattering, the light scattering from this green light. And then if you see the one on top over here, that's the automated liquid injection system that can tell, basically we can tell this system to inject certain amount of inducer once the cell density reach certain amount. So this is sort of like the cheaper alternative to a bioreactor kind of thing. So we're working on optimizing this process at the moment. So I know I'm running out of time, so I'm just gonna looking at just show you briefly what other target we are working on. So we also have this PFU polymerase that we're working on. This is the the lead on that project. We are able to get enough purified PFU to support 1,200 PCR reactions just last time. This is sort of like market value is about $1,500 or so, right? And then we also working on the nanobody. This is the anti-GFP nanobody. This is the student who are actually on the lead of this project. And I want to give a quick plug on the upcoming antibody hackathon, which is gonna take place on August 7th. This is the nanobody, anti-GFP nanobody is just one of the several interesting projects. If any of you interested in participating in the hackathon in August, you can either scan this QR code right now, and then you can register there. But you know, we did what we're doing here. Our project, our role in the hackathon is to develop affordable antibody engineering classroom activity. So we're still in preliminary work, but certainly there are a lot of things we can optimize. Of course, we are working with various fluorescent protein in an effort to create that affordable antibody engineering classroom activity. We choose our antigen. We choose fluorescent protein as our antigen because it's cheap and we get it's easy to make. You can make it in house, and you know there are a lot of variants. We have quite a bit of them. Some of them are from Biotech, the kiwi, the cherry, and peach, and some of them are actually we purchased that. Sandy helped me purchase this, and some of them we start working on that. We're able to purify some of them, but yeah, you can see right here that's a video that of GFP purification. But anyway, I think that's our my last slide here. That's our team, me and Bruce Ness, the co-PI in this grant. We have Wendy Wooten, who is our champion in the Reseda High School. She is the highly dedicated faculty at Reseda High. And then we have Dr. Wendy Johnson, who is also here in the talk. This is our close industry partner. Whenever we have trouble, we run to her like help, and then she always provide help. So anyway, so that's our first cohort, happy student over here. And if you have any question, reach out to me or connect with us. We have our Instagram as well over there. So I think that's all I have.
Thanks, Aaron. Sounds like you've been busy. I mean, everybody has, I know. All right, so I know Joseph and Kissaou have been busy answering questions in chat, but I think we still have a couple left. And I'm going to see if I can post the link to the survey here. All right. Oh, great. We do have a survey that we would really appreciate that is everybody finishing before they leave today. The surveys really help us and give us data and give us really good feedback about what we should be doing and help guide our work with the community. Okay, so let's see, some questions. I know there was one that I saw that hasn't gotten answered yet. Okay, so let's start with one. I guess this is Kissaou asking Aaron. If
yes.
Yeah, Aaron, Kissaou, do you just want to ask him?
Yes, when you show the picture that when you use a Sonication versus Freeze-Thaw, you saw that Sonication has more, I would say, efficacy or is more potent. So don't you think Freeze-Thaw may affect the activity?
Yeah, that's a good question. Yes, generally Freeze-Thaw is regarded as the sort of like more gentle lysis compared to, let's say, Sonication or microfluidizer or even detergent lysis. Right? Detergent lysis is just destroy the protein, right? So so generally it's gentler, milder way, but then as a result, you don't get as much cell that lyse. So most of the Taq polymerase are still inside the cell. So that you can see from the gel that I showed earlier, the Freeze-Thaw, there's very less proteins in the same amount of volume, even though we standardize it based on the volume already. So where Sonication utilized more. So that may be why we get more activity.
Okay.
Yeah, I remember freezing things in liquid nitrogen and then thawing them to lysozyme release proteins. So it's a method I'm familiar with from a long time ago. There was a question for you, Aaron, from Michael Fuller about the number of students that are continuing from the internships and continuing on at LA Pierce. I know you had a slide with the where different students came from for your program, but I forget how many were interns.
Yeah, I think four out of the 20 that originally signed up for the course going into our biotech, the first biotech course. So yeah, we try to track it. I have their email, but of course, I'm trying to get them into making LinkedIn, but that's been a struggle so far. So I'm working on it.
And high school student, you're not supposed to make a LinkedIn account if you're under 18.
Oh, that may be why.
Because
okay.
that might be, yeah. I mean, you can always slide, but anyway. Let's see. I have a question for Joseph, and that has to do with the virtual mentors. So I was wondering, with your virtual mentors, what you have to do to screen those, since they're college students working with high school students. Do you have to have some kind of parental permission, or what do you do to set those up?
Well, they're typically not college students. Usually these are professionals. So a couple of them were from Thermo Fisher, like a local biotech company. A few were professors at the University of Texas and so on. We don't screen them. There is no formalized process for them to work with them. I have students reach out to them to make initial contact, and then they just meet over Zoom in order to have their virtual mentoring. We initially wanted them to come into our campus and work with our students directly and have sort of one-on-one, face-to-face, I guess I should say. But most of them were so busy, the only way we could get them to commit to working with students was over Zoom. So we don't have a formalized process in place. And as far as I know, I don't think it's required for our school district.
No, I mean, it sounds like a really great idea. I just remember mentoring and having to go to the police department to get fingerprints. So,
wow. No, so that's not a requirement for our school district, as far as I know. So students just meet with them informally on Zoom.
Seattle School District may have gone a little bit overboard when they first started some of these programs. I don't know. Okay, I want to remind everybody about completing our survey.
I have one more question for Joseph and possibly Aaron. Do you have data about whether any of your students wish to do or are able to get jobs right out of high school, like jobs in biotech labs and industry?
That's something we are working to track right now, because this is our very first cohort. This is also interesting because a lot of biotech program in our area, we have like a mix of students. Some are in the transfer path, some are actually trying to get into employment, right? But in our cohort, when I did the survey, just asking them like, what do you want to do after the program? Most of them indicated they want to transfer. So which is very strange to me. There are maybe only about 3 that are actually looking for employment right after, and they are not the high school student. The high school one are the one that's they want to go to bachelor degree. So yeah, very interesting. So, but we'll see. I can give you more update next year, I guess, once I get the data.
Joseph, would you have any data about that question?
Yeah, so most of the students who complete the level 1 certificate in high school, almost all the students from my campus are going to 4-year institutions after high school. And what I hear, and I haven't had a good way to collect this, is anecdotally, a lot of them will reach out and say, hey, I got into a freshman research lab, or I'm working as a lab technician in a research group at whatever 4-year institution I'm in. And a lot of them go and get that 4-year degree. They pair that with that level 1 certificate. So they have both the 4-year degree and the level 1 certificate. And I've been running my dual credit program for going on 15 years now. And we have tons of students who are in biotech industry at all kinds of different levels. So to answer your question directly, very few of them, or I should say none of them, to my knowledge, graduate directly from high school and go right into biotech. Almost all of them go to a 4-year institution, get a bachelor's, pair that with their level 1 certificate, and then get into biotech that way.
Thank you.
I just reshare my screen. Somebody asked about the QR code. So
Thanks, Aaron. Are there more questions? Go ahead and jump in and ask. Well, that's great. Just remind everybody about the survey, and thanks, Aaron, too, for giving us a plug for the Antibody Hackathon. We're going to have some really great projects this summer in August. We would love to have students, faculty, industry members participate. It's a really good time. People work in teams to create things. That's what hackathons are all about. It's a short, intense period, but we have a lot of fun projects that come out of it. And you can find, if you go to Innovate Bio and look at events and scroll down to the very end, it's towards the end. It will be there. And I think, do we have one more question here? Let me just check. Oh, we just said, I guess that's just Joseph's email. Any more questions? No?
Well, thanks again. And I put my email address in there in case anybody had a follow-on question for us or wanted to collaborate. Please reach out. Kissaou and I would be more than happy to have another conversation with you guys.
Yeah. Well, thanks again to our presenters, Kissaou and Joseph and Aaron. And we'll have your presentation up on at Innovate Bio by Monday afternoon.